Hi guys, it's Mr. Y. Today we're going to be going through the different types of evidence for evolution. Now, I originally designed this lecture to go along with an evidence packet, so if I gave you the packet, you might want to just jot some notes down on there. We'll work through it pretty much the same way as it is in the packet. Otherwise, just take notes as normal on a notebook or a loose leaf piece of paper. So if you don't know, if I didn't give you the evidence packet, don't worry about it. I would have given it to you already. But if I did, you can pull that out and look at some of the same stuff we'll be going over here. Just make sure you do record some of the notes. So the four general types of evidence for evolution that we're going to go over today include fossil record evidence, comparative anatomy evidence, embryological evidence, and biochemical evidence. Your responsibility is to know the differences between those and some specific examples for all four types. All right, so first we're going to deal with fossil evidence. Now with fossil evidence, I've talked in the past about different types of dating systems for learning how old fossils are. And the first and most obvious one that scientists came up with was the law of superposition. That's the idea that in most cases the fossils we find deeper in the ground are in fact older than the fossils we find near the surface. So it's a relative dating system, meaning it doesn't give you an absolute hard line to say this fossil is so many hundreds of years old or this fossil is so many thousand. All it tells you is this fossil is probably older than this fossil. And keep in mind, fossils can be in the form of imprints, molds, casts, petrified remains. They can be preserved in amber. If you ever watched Jurassic Park, an example of that would be that little bug they found in the amber, or even frozen in ice. Petrified remains is when you replace the organic stuff with minerals, so it almost becomes stone. Casts and molds like footprints and imprints, things of that nature. Keep in mind, coal and oil are in fact organic fossils. They're usually marine creatures or usually plant creatures. And so it's oftentimes the case it's actually a marine plant that makes up coal and oil fossils. So every time you're actually putting in gasoline, you're actually burning refined fossil fuels. They call them fossil fuels for a reason, because they actually are made up of fossils. And then I've spoken before in a different lecture about half-life. And remember, half-life gives a confirmation, an actual way to calculate the age of fossils within a pretty certain degree. And most of the time, half-life generally supports the major trends of what we already knew about Earth's timeline in terms of the idea of life starting relatively simply and then growing more complex and complexer over time. Yes, you do have a few mass extinctions here and there, but in general you start seeing those basic trends of the timeline of the history of Earth that we covered in a different lecture. So again, I've shown you this slide before, but it's just to emphasize the idea of the law of superposition, the idea that the deeper in the ground we dig, the farther back in time we see. And that's not always the case. Things can screw up these layers, like an earthquake or a volcano can screw up what layer you find where, and then it gets really hard. And that's where things like half-life and radioactive dating have been really, really helpful to sort out exactly how old things are, no matter what has happened to the layers of sediment themselves. So I'm going to continue on with comparative anatomy evidence, and keep in mind anatomy refers to an organism's structure. So how does it actually physically look? And when comparing different structures from different species, we can find that in some cases we see cases of what we call homologous structures, where it seems that the structures themselves have the same evolutionary origin, but they're not necessarily being used for the same function. So for instance, this picture I love, it's a great picture. This shows the animal appendages of most vertebrates. Now keep in mind, vertebrates are things with backbones, things that have an internal skeletal system. And you can see in all cases there's actually a pretty common setup. You have one bone branching out from the trunk, followed by another two bones, followed by the manipulative bones, the wrist bones in our case, and then 5 pharyngeals, 4, 5. And then again, the same pattern, 1, then 2, the wrist, and 1, 2, 3, 4, 5, and then 1, 2, the wrist, and then 1, 2, 3, 4. But you can see where this probably at some point had a 5th right there. And then again, same pattern here. It's hard to see the second bone, but it's really small here. And then 1, 2, 3, 4, 5. So nobody would argue that each of these creatures uses the appendages in the same way. Humans use it for grasping, cats for walking, whales for swimming, and bats for flying. But if you actually look at the internal structure, it's like, well, yeah, there's a very similar pattern of the way the bones are organized here, meaning they probably have the same evolutionary origin. They've just been evolved in towards different functions at different points in time. This is an example of homologous structures. So keep that in mind. Homologous, same origins, but different functions. Now, sometimes the functions can be very similar, and this is an example that these are still homologous structures. Again, same basic evolutionary origins, but they're all used for grasping, even though you can see there are some obvious differences. Most of these have 5 pharyngeals, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, and then 1, 2, 3, 4, and 5. And they're all used for grasping, but keep in mind, it's not so much the function we care about. Is the origin the same evolutionarily speaking? Did they evolve the same 5 pharyngeals at the same point, or did it evolve separately? That's going to be in our next key term. So all these guys use it for grasping, and you can see they have very similar setups, leading us to believe they have very similar evolutionary origins. Yes, there is some subtle differences here and there, but overall it's very similar considering the breadth of the animal world. So the opposite of homologous structures is this term, analogous structures. These are structures in different species that have the similar function. They have very much the similar purpose in that species, but they are so different, biologically speaking, so different anatomically, that we assume they actually have different evolutionary origins. So let me give you an example of this. So here we see 4 examples of wings, which means we can look at these and say, okay, every one of these creatures can use the wing structure it has to fly. Now the question becomes, well, do you think all of these evolved from the same common ancestor? And the answer is no, probably not. The first structure here, this little insect guy, has absolutely no bones to speak of throughout his entire body, let alone in the wing. His wing is made up of just membrane and veins. This wing here, from a reptilian dinosaur, has a very similar setup to some others you might see in the dinosaur kingdom, but it looks nothing like the other two in that there's no feathers, and the wing itself is actually still made of a membrane, so definitely not feathers. And you can see, if you actually look at the pharyngeals, you have 1, 2, 3, 4, and then the 5th one, what we would consider to be like our pinky, is greatly elongated, and that's where most of the membrane fits in for the wing. And then we have here, obviously, a bird, again, 1, 2, and then 3, 4, 5. And birds, of course, have feathers that looks nothing like anything else that had come before that in terms of flight. And then here you have a bat. And a bat, if anything, it looks more similar to a dinosaur than a bird. And of course, there's no feathers, but if you actually look at the way the membrane is attached, it's attached between the arm, what we would consider the arm bones, and the different pharyngeals here, here, and here, unlike the dinosaur, which has most of the membrane attached just to that last pharyngeal in the arm. So all these have the same purpose, the same function. They're all used for flight. They're all capable of flight. But because they are so anatomically different, we assume they have different evolutionary origins. These are analogous structures, not homologous. Remember, homologous means same origins, not necessarily the same functions. Here is another example of analogous structures. Actually, there's 2 examples. One right here, a gentleman has an eyeball. And right here, this creature also had an eyeball. This is the eyeball of a giant squid. And it looks very similar, actually, if you look at it just in very superficial level, to a human eyeball. Here's a blow-up of the anatomy of the two. And they have the same basic function, but there are some minor differences that lead us to believe that, yeah, the squids, the cephalopods, the invertebrates, actually evolved the eye independently of the vertebrate eye. So they actually have just as good eyesight as we do, but because of slight anatomical differences here, and the fact that squids, octopi, thing like that, they have no backbone. We assume their eyes evolved independently towards the same general purpose. That is to be able to have really good depth perception, to have really good color vision, to detect movement. That's basically what the vertebrate eye does. Again, this is an example of an analogous structure. It's considered analogous because we have this guy down here, the whale, which is a mammal, and you have this guy up here, which is a fish. But you can see, while they are very much different species, they have the same basic body form when it comes to swimming. The form itself would be considered analogous. It's different origin, same purpose, to streamline the body shape while this creature is in water. And again, one is a mammal, one has the ability to produce milk, give birth to live young. The other, being a fish species, lays eggs, doesn't produce milk at all, doesn't need to breathe in oxygen from the atmosphere, can breathe through its gills. So the analogous structure is the body form. It's different origins, different evolutions, but same basic purpose, same function. Again, here is a case of analogous structures as well. This is kind of called convergent evolution, where we have multiple species. They kind of converge onto a very similar answer to a very similar type of evolutionary problem. Each one of these species, and I can show you two at the top, but I can't show you the shark because it just wasn't in the picture. They all evolved very much the same type of body form and the same type of, if you look right here, teeth. And all of them are very fast-moving, high-metabolism swimmers, and they tend to prey on smaller fish. And you can see shark, which actually falls under its own category of fish, the dolphin, which is a mammal, and this thing was actually considered a lizard. So these things actually evolved very similar forms from different needs, but because their needs were very much the same, they evolved very similar solutions. All right. Going on with another key term in comparative anatomy, and this is vestigial structure. Now, a vestigial structure is an organ that serves no useful function, but it does resemble a functional organ in other species, and humans actually have a few of these. Humans have ear muscles, we'll talk about those, and a third eyelid. Now, this guy here in the bottom corner is actually a functional third eyelid in a mammal species. Our eyelids don't do this, and this third eyelid starts here and actually closes this way, and you can see it's fully closed here, and it works as an additional protector for the eye that's actually transparent. The animal can see through this. And you can see it here in this bird picture. The bird here uses it because birds have to travel at high speeds, and oftentimes it's a protection system against wind, debris, and actually insects hitting their eye. Reptiles, like crocodiles, have a third eyelid here. In humans, this is a vestigial structure because it serves no useful function and it's not in any way functional itself. It actually exists right here on your eye. Occasionally it can become inflamed. If you ever had a dog who had their third eyelid inflamed, it happens more often in dogs than it does with people. The veterinarian would have called it something like cherry eye, and it gets really big and really red and it becomes really painful. So it's not functional in humans and a few other primates. It is functional in other species though. So this is the idea that there was a structure that was functional at some common ancestor and then evolved to continue to be functional in some species and non-functional in others where it wasn't needed. Oh, and this eye right here is from a camel. Now, if you think about it, why would a camel need a functional third eyelid? Well, where does a camel live? It lives in the desert. What's in the desert? A whole lot of sand that can easily get into the eye and damage the cornea. So it needs to have additional layers of protection to stop blowing sand from getting in there. So in keeping with vestigial structures, a lot of times you actually see vestigial structures more apparent actually as the creature is an embryo or still in the developmental stage while inside the womb. What you see here, three pictures, A, B, and C, of actually a horse's leg developing inside its mother. And what you can see in the very early stages, this thing actually has 3 toes. And as you can see, as this thing gets more mature, the toes on the sides actually become much smaller, more vestigial, that is to say more useless, and they actually retract, and you can see the beginnings of what you would consider to be the horse's hoof right there. In a true anatomical picture of a horse's leg. You can see this is with, all the skin and stuff right here, but if you actually remove the skin and the hoof and all that stuff, the vestigial structure of their 3 toes is on each side, and here's one of them right there. So vestigial structures oftentimes are not functional when we call them vestigials, but in some cases they can be functional in other creatures. Well, if you think about it, what animal do you know actually has 3 toes? Well, there's a few things. Things like rhinos have 3 toes, and a few other big land walking creatures that has 3 toes as well. In whales, we see a very clear example of this with their bone structure, because if you actually look at the skeletal system of a bone of the whole whale, you actually find this one little piece way up here, and they circle it down here. This is actually a pelvis bone. Now, the purpose of a pelvis bone in every single land animal you can find of is to be the connection between the spinal cord and the legs. Let me draw a little leg here. But obviously, whales don't have legs. So why would they ever need a pelvic bone? Well, the answer is probably they don't need the pelvic bone. That's why it's so small. But their ancestors probably needed the pelvic bone to connect to legs, and the legs became vestigial and then eventually lost formation due to the need for a swimming formation. But the pelvic bone remained because it was not in the way per se. It's just greatly reduced in size. So the pelvic bone of a whale is actually really, really small compared to their size, and it doesn't need to be big because it has no useful function. But we know pelvises in any other land creature actually is fairly large compared to their sizes because it has to connect and support the legs to the spinal cord, so those can be walking upright, or in at least a 4 appendage position. In humans, another example of this is the cocci, the very, very bottom bones underneath the pelvis right here. And these are what we call the tailbones. They are very, very small, and they break very, very easily. They're one of the easiest bones to break if you're not careful. And they serve absolutely no useful function whatsoever. But they do seem to resemble what? They seem to resemble tailbones from other primates and even other mammals in some cases. So it's thought that cocci, the tailbones, is actually a vestigial structure from primate origins. There are many other apes that have the same setup, like gorillas and chimpanzees, and their cocci are just as vestigial as ours because they don't have tails. So I mentioned this one before, the ear muscles of humans. Some people can move their ears just a little bit, some people can't move their ears at all, but nobody would be able to say they can move their ears to the degree that something like a rodent, like a mouse or deer or horse or a canine, can move their ears. We do have ear muscles though, and they really don't have a useful function on us. Well, then what are they doing there? Odds are they came from a common ancestor at some point who did have a use for those ear muscles. Same thing with the third molar. Most people have to have their third molar, what's known as their wisdom teeth, removed. They usually don't serve a useful function, but they probably did at a common ancestor at some point. The tailbone, which I've mentioned before, the cocci, again, they serve no function in most of the great ape family, gorillas, orangutans, chimpanzees, and they certainly don't serve a useful function in humans, but they do seem to be very similar in origins, just anatomically speaking, to other creatures that do in fact have working tailbones. And actually, if you look in your intestines, there's one portion of your intestine right here. This is called your appendix, and people can live without their appendix. It serves no useful function. All it can do really is get inflamed, get infected, and have to be removed. So that's a pretty common surgery to have your appendix taken out if it does become inflamed, and it doesn't do any lasting damage to have it removed because it's vestigial. It doesn't serve any useful function in the human anatomy. Another example of this, called Darwin's tubercle. It exists right here on the ear. Here is another example, and you can see it's kind of similar. If you actually were able to fold the membrane of your ear outward a little bit, it would kind of almost be exactly at this point here. And so about 10% of people have them. You can usually pick out, if you feel it, like a little dimple right there, if nothing else, to see exactly where it would be. It's not too difficult. Some people it's really not apparent though. But you can see if you actually were to fold out the membrane of the ear a little bit towards the outer edges, you would see something more like this, which is very similar to other primate species. All right. Now, let's talk more about embryological evidence. I mentioned a little bit about that with the vestigial stuff, but I want to go more in depth. Now, scientists learned for a long time, and Darwin even knew this, that if you looked at the early stages of development of most creatures, it gave you a really good insight into common ancestry. So, for instance, when you actually looked at the embryos of stuff like fish, reptiles, birds, and mammals, they all had some similar features at the very early stages. Now, they looked a little different, and I admit these are diagrams, and I will show you some more accurate pictures in a while. But here, they all possess this area, which we call gill slits. Now, these are not gills. These are not functioning gills. The gills are an organ that exists beneath the layer of the inside layer of, whether you want to consider it gills or scales or flesh, but these are gill slits. Now, in fish, they become the slits that actually cover up the gills. In reptiles, they actually become nothing. They're just part of the neck. Birds, nothing either. And in humans, they actually become part of the inner ear. So they might start off looking very similar, and then they develop into completely different structures as the creature grows into maturity, as it gets ready for birth. Again, scientists, even before Darwin, noticed that at the embryological stage, all these creatures, even though they're very different species, all had a tail. Now, of course, fish can have tails, but usually it's a membrane, and the membrane would be added on here later on as it matures. The reptile, of course, does have a tail. Birds have a tail as well, but most of the time what we see is the tail is actually their feathers. And humans, of course, don't have a tail, so this becomes part of the cocci, the vestigial structure I mentioned before. And again, they look very similar at this point, so where if I didn't have labels on there, it would be very difficult for you to tell me which one was truly which was the human, which was the bird, which was the reptile, which was the fish. Here is 3 different pictures, all entirely from human embryological development at different stages. So the top one here is at 28 days, the bottom one here 40 days, and here is 46 days. And again, you can see the tail right there and the gill slits right here. And then you can see as time goes on, the tail becomes more and more recessed, more vestigial, and the gill slits begin to disappear, and actually the skin starts to cover it up, and what was the gill slits actually becomes part of the inner ear, which would be around here. All right, so quick question time. One of these creatures they're both early developmental stage creatures. One of these creatures is a human, one of them is not. My question to you is, which one do you think is actually the human? So I'm going to now show you a few slides of progression as these things develop in the embryo, and we'll figure out the answer. So again, the question is which one, the top or the bottom, is the human? Now you can see, obviously by the end, they're very different. But in the beginning, it's very difficult to actually make a guess on which one is which. Obviously, by the time they get to full developmental stage, you can tell which one is the human and which one is the chimpanzee. But at the very early stage, almost impossible unless you really know how to study the skeletal structure, which one is which. Actually, we found that chimpanzee and human intelligence actually stays the same. They're parallel for about the first 2 years of life, meaning if you take a chimpanzee that's about 1 year old and a 1 year old human, they have about the same intelligence level. It's at about 2 years old that you actually see a difference in the intelligence level between humans and chimps. All right, moving on to comparative biochemistry, and these are just a few examples. ATP, we mentioned that last semester. Remember, ATP was used a lot of times when we were talking about photosynthesis and cellular respiration. It was the whole point of cellular respiration. ATP was energy. It was the cache of the cell. And the simple fact that every cell in every organism that is made of multiple cells, so plants, animals, funguses, protists that have multicellular systems, they all use the same type of energy molecule. They all use the same exact cache, meaning this is evidence of evolution. This has probably a common origin. The fact that all cells use this as energy is a sign that the cells themselves originally started off and then diverged into different species as such, but they all started off using this. They didn't co-evolve this. They didn't come to the same solution together. They just started with this and then evolved after that point. So even bacterial cells will use ATP. And I'm talking bacterial cells that have, they're just one cell big. They don't have a nucleus, but they'll still use ATP as an energy source. And if you think about it, if you want to get down to even more details, RNA and DNA, the fact that all life passes down information using the same basic structure of either RNA or DNA, and most of the time it is DNA, but a few cases of very primitive life forms, very simple bacteria will use just RNA. But the simple fact you're using the same basic structure, the same basic system, is evidence for common ancestry. So the energy source particle is the same in all cells. The information particle, the information medium, the DNA, is the same in all cells. All of this leads to the common idea that life on Earth evolved just one time, and it got these two very early evolutionary traits early on of how to pass down information and what energy source to use, and then that spread throughout the entire tree of life. Keep in mind, many genes, many segments of DNA that code for proteins between species are actually so similar you can interchange the two. If you actually took a certain gene from one mammal and put it into another, it would function the exact same way as it did in the original. And scientists have done this before. And they've had some really weird experiments because of that, where they've taken genes from a mouse and put it into a different type of mammal, and it's functioned the same way. And even if the species are vastly different, even if they're not two mammals. So one of these experiments where scientists interchanged genes came from a gene called PAX6. And what scientists did is they took the PAX6 gene from a fruit fly and they put it into a mouse, and they did this at a very crucial stage during the development of the embryo when the mouse was trying to develop its eyeball. And what they found is the mouse came out perfectly fine. It developed a perfectly formed mammalian eye. And then when they took the PAX6 gene from a mouse and then put it into the insect at the same developmental time when the insect was developing to maturity, it developed a perfectly normal insect eye, meaning PAX6 has the exact same functions in mammals as it does in insects. It starts the development of the eyeball, no matter what kind of eye it is, whether it's an insect eye, which is vastly different than our own, or mammalian eye. And like I said, you can look at the anatomy here and you can see these two eyes operate in very different ways because of the anatomical structure. We truly have no idea how insects actually perceive the world because their eye is so anatomically different than our own. We can only make guesses at it. But the same gene, PAX6, actually triggers the development of the eye in both mammals and in insects, thus leading to more evidence for evolution for common origin of that gene, PAX6. And it is a very crucial gene we found out in almost all animal species. So the PAX6 gene triggers eye development in mice and works just as well in all mammals. They've actually changed it between mammal species as well, and it worked fine, and some insects. Now, here's where scientists got a little weird. They took the PAX6 gene from a mouse, again, the mouse species, and they put it in the insect, but this time they inserted it in the wrong position. They put it in the wrong part of the body on purpose, and they put it onto what would be called its antenna, and they actually got an insect eye to grow on the antenna. And they found when they dissected this thing later on, there were actually some nerve connections going to the central nervous system of the insect, meaning we don't know how functional it was, probably not all that functional, but the information this eye was receiving was somehow being transmitted to the nervous system. Then they did the same experiment again, and this time they put it on the insect's legs. I guess you would consider that to be its knees, but insects don't have knees. But you can see the optic eye here. And again, there was nerve connections going back to the central nervous system. And again, this is all using a mouse's PAX6 gene. And you can see here on the antenna, the extra eye right there. So genes themselves are often very similar across a wide range of species, such that even if you replace them across species, they serve the same essential functions. Not always, but in some cases, it's almost identical. 100% of the code is exactly the same. And here's some more pictures from the original study that was doing this back, I want to say, around 1998 or 2000. You can see the extra eye here, here. This is blow up at the top picture. And here's the antenna one. This is blown up down here. So again, yeah, these were the result of the scientists taking the PAX6 gene from a mouse and then putting it into a fruit, an insect at different locations. The insect actually developing these optic eyes based upon the mouse's genes. So let's go over a little bit about the evidence for evolution that Darwin knew and some additional stuff that Darwin didn't know during his time. First off, as I said before, Darwin's history came as a geologist, so he knew a lot about the fossil record. He didn't know about half-life, but he definitely knew about the law of superposition, and he knew that if you looked at the fossils, you saw apparent changes in species over time, and he knew that a lot of species went extinct. They didn't appear to be alive anymore, and that some species had grown larger, some species had apparently grown smaller, and that there were several cases of species just dying off. What he didn't know, the half-life stuff. He didn't know anything about radiation or how to actually calculate how old the Earth really is. He did do a few estimates based upon his fossil studies, but those were just general guesses. They did push it back into the Earth being a couple 100 million years old, but we know from half-life that the Earth is about 5 billion years old, based strictly on radiation. But so he knew nothing about that. But he did know that species were changing over time, some were dying off, and new species were evolving. Embryologically speaking, he had microscopes, so he had access to look at embryos, and so did other scientists. He could see things that would be vestigial in many cases. He would, for instance, see the bumps in the embryos of snakes that would, in other species, grow into legs, and then snakes stay very much vestigial and never develop into anything. So he knew a lot about the embryos of different things looking under microscopes. He didn't know about the biochemistry. He didn't really know because scientists hadn't really discovered the purpose of DNA or genes or stuff like ATP or proteins. They only knew that somehow parents looked similar to their kids and kids looked similar to their parents, that somehow information was being passed on, traits were being passed on, not identically, but there was similarities. Comparative anatomy, he knew all about vestigial structures in creatures that apparently had no function, but if you actually examine them very closely, they looked similar to functioning structures in other creatures. So he knew a few of those. He didn't know as many as we know today, but he knew definitely about a few of those, such as the ear muscles, the cocci, things of that nature. And he didn't actually know how speciation worked. He knew it needed reproductive isolation, the idea that you had to have species get separated somehow and not be able to interbreed for a new species to evolve. And he knew that the environment pushed for better adaptations to help towards survival and or reproduction, but he didn't actually know how it went about through the entire process. And finally, he knew about the process of artificial selection, which we'll talk more about in the next slide, the idea that people can artificially select for how species tend to evolve. You see this in the case especially of, and this is where Darwin got his first insight, dogs. He knew that if you kept breeding smaller dogs, eventually you get smaller and smaller and smaller dogs till you have what we would consider today to be the little lap dogs, the chihuahuas, the shih tzus, the things of that nature. And he saw the same thing in horses, pigs, farm plants, any type of livestock you see on a farm. He knew all of that was artificially being selected for to serve human needs. And he simply said, okay, if humans can do that, why can't Mother Nature do that, just with the idea of being geared towards survival and reproduction for the species. So this slide I'm actually going to go over again in one of my next lectures, but I'm going to go through it real briefly here and we'll talk more about it next time as well. So again, Darwin didn't really know how speciation happened. He knew it had to have reproductive isolation. He just didn't know necessarily how you always got to reproductive isolation. He probably thought of geographic isolation, the idea that you have species somehow getting separated. Thus, you had the Galapagos Islands, which were separated from mainland South America by a pretty big distance, and some species probably got blown out there thanks to things like hurricanes. And so because they were geographically isolated from the mainland, you then had reproductive isolation of a population, and that can lead to new species. Behavioral isolation that means where the way the animal behaves is slightly growing different and different to where you have two different types of populations and thus leading to reproductive isolation. An example would be like mating dances in birds. If birds if you learn anything about documentaries on birds, you'll learn they all have different types of mating dances and mating courtship rituals to gain a partner. And so if you have a simple behavioral isolation where one population does a slightly different mating dance than another population, that can lead to reproductive isolation. And temporal isolation. So if you have, and this is kind of a mix between the two, if you have a species that's nocturnal, that's only active at night and not active in the day, and another population that's more active in the day and not active at night, and I'll give an example of this in my next lecture, this can also lead to reproductive isolation because obviously some species will be awake while other species are asleep, and then vice versa. And again, all this can lead to reproductive isolation, which leads to speciation. And speciation itself is just the idea of that from a common ancestor, you see reproductive isolation give rise to two new species. Remember, the definition of species is organisms that have the ability to mate and produce fertile offspring. So species A and species B would be considered divergent enough to where they are no longer the same species. They can no longer interbreed between the two. And when true speciation occurs is somewhat debatable. Again, there's kind of a gray area here where maybe they could still interbreed, but at some point it doesn't work anymore. It doesn't really work within the genetics of it. So when that point is debatable. It depends upon the species you're talking about, and some species are still diverging, and some species can actually do interbreeding between what we would call different species. I'll give examples of that in class. But this point here is not exact. It's kind of wishy-washy depending on what species you're referring to. But if you have a clear, distinct set of species, like, say, a giraffe and a rhino, those are obviously very distinctive species. They cannot interbreed. They cannot produce fertile offspring. We do know they have a common ancestor because they are, in fact, vertebrates. They do all have backbones. They're mammals. They have hair. Yes, rhinos have hair. They have eyelashes. And they produce milk for their young, and they give birth to live young. So they do have a common ancestor at some level, a mammalian ancestor, but at some point their common ancestor diverged into different species that eventually gave rise to what we know as the giraffe and the rhino. So that's going to wrap up this lecture. I know it was a long one. Make sure you ask questions in class, and there will be one more lecture on evolution, so I'll see you guys in that one.